Can you take your seats, please? We are about to start. Welcome to the second session of the morning. This session is specifically giving you a brief overview of what the EIC Pathfinder can produce in terms of exciting future technologies. The first half hour will be dedicated to medical and neuro-technologies, and the second half hour will be dedicated to energy and environment. But before that, and before introducing each of the speakers in each session, we'll show you a video. We've come about the celebration of the 30 years of PhET, and that will give you a glimpse of what PhET can do or what PhET has been able to produce in the past 30 years. Can we see the video, please? And apologies for those of you that have seen the video already.
Happy birthday. Wow, 30 years already. Gifts are better given than received, so this gift is for you. Our modern world is full of gifts. Take her smartphone that enables more than just calls. It helps us explore, connects with friends, notifies our health and virtual worlds. Gifts like these can bring us health, happiness, and comfort. Next generation technology for faster analysis, accurate diagnosis, personalized treatment for a quicker recovery. Research on future and emerging technologies is unwrapping our future potential. It works by building research teams across different areas. Biologists with computer scientists can create digital monitoring devices. Physicists and designers are improving human-computer interfaces. It all starts from a small group of professionals with a shared vision. Like making machines the size of molecules, batteries that never need to be charged, or virtual worlds that feel entirely real. And it grows, passed from person to person who share these visions, transforming them with their knowledge, gradually turning the dreams into reality. Like mapping the entire human brain, prosthetic limbs powered by our minds, smart clothes to help us exercise and rest. Exploring courageous ideas is not without risk. But creating green fuels, connecting brains and constructing quantum computers comes with big rewards. As we mature, we get better at these things, more able to learn from failure, more likely to harness success. This gift brings a better future closer to reality. A future we don't just dream about, but one we can actually build. Designed by connecting research teams across Europe, commanded by intelligent speech recognition, assisted by safe and secure artificial intelligence. When turning 30, there's much to celebrate, but a lot more to come. For 30 years, researchers funded by the European Union have been working on future and emerging technologies. We call it PhET. One of the first dedicated research programs in the world to take a dream, a vision, for how good the future could be. PhET has planted the seeds for many of the exciting technologies we see today, turning science fiction into reality. A person from the past might call it magic, but it's not magic, and it didn't happen in a day. PhET is celebrating 30 years of courageous research, and its gift to you is knowledge and what we do with it. Together, we are making the future brighter, better, unbelievable.
Thank you very much. And now we go through the three projects that will give you a glimpse of what PhET has been able to do in the area of medical and neurotechnologies. The first project is Nebias, and the speaker will be Giulia Dominigiani from the Transnational Neural Engineering Lab in the École Polytechnique Fédérale de Lausanne. He's accompanied also by the originator of the idea, Professor Silvestro Micera. Giulia, the floor is yours.
So good morning, everybody. So with this brief talk, I'm going to give you some insight on the Nebius European project, which aimed at contributing to the current quest for a bionic hand. Let's make a step back. Our hands are formidable tools characterized by a large number of degrees of freedom and an incredibly high amount of receptors. And that's what makes them capable of both fine manipulation and powerful grasping. So for this reason, hand loss is an extremely disabling event, and therefore the humankind have always tried to restore dexterity with the technologies available at their times. Nowadays, current state-of-the-art active prostheses interface with the residual muscle to allow direct volitional control, but what's actually lacking to develop a Star Wars-like bionic hand is rich sensory feedback. Indeed, real-time and natural sensory feedback could improve control performance and increase the functional impact of prostheses in activities of daily living, which in turn will also increase the full acceptance of this device. Now, when you want to provide natural sensory feedback, you have to interface with the nervous system. And there are several ways to do that. One of these is to interface directly with the brain, stimulating, for instance, the somatosensory cortex, or focusing on peripheral nerves. Now, in general, when possible, one should always try to stay as peripheral as possible, because it wouldn't make sense not to take advantage of the existing neural structure and their actually intact pre-processing capabilities. So for this reason, for us in this project, bidirectional neurocontrol hand prosthesis is a device which is able to stimulate sensory nerves to provide sensory feedback, while actually also extracting motor commands from the motor nerves. But then, when you really want to interface practically with the nervous system, with the peripheral nervous system, there is a bunch of electrode types from which you can choose, and these are characterized by the degree of selectivity, which is how well you can stimulate the nerve and the different fascicles, and a degree of invasiveness. And it seems that intraneural electrodes, and in particular the transversal intravascular multi-channel electrodes, represent a good trade-off between high selectivity and reduced invasiveness. So for this reason, they have been the technology of choice for the first short-term implant in a transvagal amputee, which lasted 4 weeks, and Professor Rossini in Roma implanted 4 of these electrodes, 2 in the median and 2 in the ulnar nerve, of once serving the amputated hand. And upon stimulation of the different channels of these electrodes, the patient was actually reporting a large variety of sensation, which could be modulated according to the stimulation parameter. And the sensation were actually reported on the phantom hand. So given the possibility to actually elicit sensation on the phantom hand, it was possible to test to what extent the subject take advantage of such information during manipulation experiments. And actually, it has been shown that the subject was able to match requested force level, and not only that, but also to notice overshoots in exerted pressure and adjust accordingly to meet the requested level, as it's indicated by the arrow. But besides this, the subject was also able to discriminate between objects of different kind in terms of shapes and compliances. And what is even actually more striking is that their performance are comparable to those of LT subjects. Now, one could say that this was just one patient and for only 4 weeks, and actually he would be right. And so for this reason, 3 additional patients were enrolled in a longer-term study, and the results obtained with this clinical trial actually confirmed the previous outcome and proved the long-term usability of this approach. Now let me show you some video. Here we have one of the patients performing the so-called virtual egg test. So basically he was able to pick and place small cubes as fragile as eggs, thanks to the provided sensory feedback. Now to you this might seem a simple task, but actually doing this just relying on vision, it's nearly impossible. But beside indoor lab tests, the subjects were actually also able to exploit the device outdoor, performing proficiently activities of daily living such as drinking, writing, and even driving a car. And this was granted by the fact that all the electronics needed could be carried in a small backpack. So now these are the achievements of the Nebius project, but I would like to conclude showing you where are we heading to from here? And in particular, we are now focusing on the development of a fully implantable permanent bionic hand, and the first implant is expected before the end of 2020. But actually, the technology and know-how on neural interfaces developed within the Nibios project will not be exploited only in the field of hand prosthetics. Indeed, with the new heart project, which is a new FAB project in collaboration with Professor Leonetti, we are actually going to stimulate the residual vagus nerve to reestablish the coupling which is missing between a transplanted heart and the autonomic nervous system of the subject. And this is what actually allows fast and healthy heart rate regulation, which is what is lacking in transplanted patients. Thank you.
Thank you very much. Now we move to the second project, is Project C-RES Space, which I have the honor of being the scientific project officer. We'll have two speakers from this project, Professor Alan Nogaret from the Department of Physics in the University of Bath, UK, and Marian, excuse me if I don't pronounce your name correctly, Giongiosi, okay, from the Medical University of Vienna. She's a medical doctor. Okay, Alan?
So the basic vision behind the CRES-Space project is to develop adaptive neural electronic which respond to physiological feedback from the body and deliver carefully timed impulses to the body itself. So basically it's a closed loop feedback system. One example of application of this is to heart failure therapy. So this basically these are videos. In a heart failure, the heart is really enlarged and doesn't pump very well. In the healthy heart, the heart is very small. One prognosis for heart failure is a loss of heart rate variability. So basically what CRES-Space is doing is to sense physiological feedback to restore the normal heart rate variability. So if your heart is healthy, your heart rhythm is not constant, it's not a regular beat. What happens is that the heart beats a little bit faster when you breathe in and a little bit slower when you breathe out. As a result, the heart rate oscillates as a function of time. So that's for a healthy person. So basically, the fast intervals correspond to the increase in lung inflation. In contrast, when you have a patient which has heart failure, the heart rate is constant, so that is bad. So we have a first parameter, which is lung inflation, which is the first physiological feedback parameter. But you know that if you go running for a jog, for instance, the heart rate will increase naturally, and this is because the gases in the blood, O2 and CO2, this gas concentration will vary. You also know that if you are drinking some coffee, your blood pressure will increase, and this will have an effect in the heart rate. So we got these four physiological feedback that we are reading with the electronics we developed. But basically, this electronics mimics the biocomputer, which is at the base of the brain, and then in the medulla. And so basically, it reads these four physiological feedback, and it applies. It controls the heart rate basically through these two nerves. One is the accelerator. This is the sympathetic nerve. And the other one is the vagus nerve. It slows down the heart rate. So basically, this is a zooming of the nervous system. And the Botzinger at the bottom are where the neural networks that we model that regulates pacing. So basically, we know what this biocomputer looks like. So each ball here represents a neuron. These neurons are interconnected via synapses, inhibitory, excitatory. You can see the SN is the activation of the heart. So basically, this is the accelerator. VN is a brake, and there is a feedback from the body into the medulla that informs how this computer should regulate the heart rate. So basically, we know what this circuit does, but we do not know how it works. So the role of the physicists, where I am within Space and others, is to have developed method to infer the parameters which allow to build quantitative models of this network. So the way, the principle behind our method is first to perform recording, electrophysiological recording, using a needle here, of networks from at the base of the brain. So you can see that the voltage oscillation of one such neuron and the current injection, that's the blue curve. And we do a sophisticated fitting procedure, which is called assimilation, from which you extract parameters. These parameters are then used to configure an electronic circuit that we transfer to VLSI, and then we make a chip. And if we have done our work properly, if we stimulate the chip the same way we stimulate the neuron in vivo, the chip will respond identically. And this is the red trace is the output of the chip, and you can see that this is something we can predict. So we can do that for neurons and also for small networks. So my last slide is we then transfer the chip inside a pacemaker, and we got some teams in the consortium. We develop sensors, including ourselves, which are tested in the lab of Mary and John Josie. And we pace animal models of heart failure. So we got some of the results we have. If we have no modulated pacing, the cardiac output goes up to 25. With modulated pacing, with our method, we increase the cardiac output up to 19%. And you know that for a patient which has heart failure, a 3% increase is a life-changing experience. So from this, I'll pass it to my colleague. We'll continue.
So just about the clinical importance and relevance of this project, that the rate of the cardiac pacemaker implantation rise with age, and population worldwide are aging. Approximately more million new pacemakers will be implanted worldwide. Approximately 70–80% of all permanent pacemakers are implanted in the patients 65 years of age or older. These patients have significantly higher comorbidities resulting in reduced heart rate variability. It is well known and proven that the reduced heart rate variability is significantly predict mortality and the lower life expectancy. Overall, the prevalence of the pacemaker implantation is estimated 0.04% between 18 to 64 years, but over 75 years it is practically 2.6%, and that means and the slide shows how is the implantation of the pacemaker is increasing yearly. So thank you very much.
And now for the third project of the first session, we have the pleasure of the company of Professor Nikolaos Papadopoulos. He's professor of the allergy and pediatric allergy at the University of Athens, Greek, and as he says, he likes rushing around the globe to speak at meetings about once a week. He'll be telling us about his third project, CURE.
Thank you very much. Hello, everybody. And we'll present to you CURE, Constructing Eubiosis Reinstatement Therapy for Asthma, a little bit cryptic title. Most of us may not realize that from antiquity, about 3 millennia, it has been infections that have been plaguing mankind, and the majority of people will die out of some infection. This has changed. It's about a century and a half where we have antibiotics, we have vaccines, also have hygiene, and it has changed completely how we see health and disease, and it has an effect. It had a great effect. You can see here that life expectancy here a century and a half ago was less than 40 years, and we're now passing in a linear way over 80 years. So have done something really very well. But of course, every action has their reaction. And what is happening nowadays is that we're having chronic, mostly inflammatory diseases, which is the major burden and the major problem that we have to face. Includes all these kinds of diseases. 8 out of 10 people in Europe nowadays die of such a disease. And of course, we pay a lot of money in trying to do something about them. Among all these diseases, the ones that have risen most steeply are allergies and asthma. They are the #1 epidemic together with obesity, and they are, we have been wondering why these diseases have been increasing so steeply. Trying to understand how the lack of these external microbes may lead to immune fitness. One possible explanation that actually is prevailing nowadays is that our internal microbes are actually affecting our health, and in fact, microbiome is the one that is mediating this immune fitness. In fact, all the studies, it is really in fashion nowadays that microbiome has been associated with up to 90% of different types of diseases. Wherever you try to find an association, you will find it there. And the microbiome is very important in many different ways, trying to help tackle global challenges like food and nutrition security, health and well-being, waste management, etc., etc. So the microbiome is really attracting a lot of attention. But what is the microbiome? It's a group of different microorganisms that live within us, and it lives in different places, and it is different types of microorganisms. In fact, we understand nowadays ourselves not as a person individually, but as an environment. We cannot live without these microorganisms. However, most of the studies that you have heard of about the microbiome are about gut and about bacteria. And of course, these are really very important. However, when it comes to asthma, which is our interest, it has been shown with many studies, which we have done some of them, that actually the majority of asthma exacerbations and different asthma presentations are related to viral infections. About 90% of exacerbation in children, over 75% in adults. And this is in the respiratory tract. So we were interested in studying the viral aspect of the microbiome, which we call the virome, on the respiratory tract. And what we did is we tried to compare health with asthma. And what we have observed that there were clear differences. That's what you expect. As I said, 90% of diseases were associated with the microbiome. Here as well, we found an association. There's a difference between healthy and asthma, a dysbiosis. But what we observed, and I think that was the important point we started our research, was what was missing were actually these little creatures. There were much less, and they were quite different. The bacteriophages. The bacteriophages are really very interesting. I mean, you would agree that this is like science fiction. It's incredible that this organism exists. Actually, it's the most populous organism on Earth, 10 to the 31st. And we have actually, this is a graph, but this is a picture. This is how they look like. And what's very interesting about the bacteriophages is that they have this predator-prey ecologic interaction with the bacteria. So if you have bacteria, then you put a phage, then the bacteria go down, the phage will go down, the bacteria will go up again. So basically, they regulate the bacterial population. And this is where we got our idea about cure. We suggested, since this balance is disturbed in allergy and asthma, and since phages are missing, what if we start putting specific phages and try to regulate this imbalanced microbiome? Will we be able to restructure this microbiome and make it eubiotic? And this is what the Project Cure is all about. We're actually taking samples from patients. We analyze them in order to understand how the specific structure of the microbial communities are. We also see at the immunological part of them. And we feed all these, we give them these to our mathematicians, and they try to construct mathematical models predicting what will happen when we put specific phages in the respiratory tract and perhaps later on in other organs. And this is some first results that we have in understanding the community dynamics, and this is it's a nice graph. Lots of colors. It shows you how the different microbes act as a community, and you see here in health. And if you compare it with the community in asthma, you will see that the community in asthma is not well at all organized, is not resilient. So we need to feed it with something in order to make it healthy again. And we use machine learning, artificial intelligence, different methodologies in order to fit the models to make these things happen. And we have built some disease classifiers. We use, as I say, these techniques in order to try to understand how one system can change into another with the intervention of phages. We have identified already some specific characteristics that can be used. Of course, there are other people that are working in separate projects and parallel in order to make phage therapy legal in Europe, because phage therapy happens in many places around the world, in former Eastern Europe, in Georgia and in Russia, because there it has been happening for many years, but regulation is not allowing yet phage therapy to happen. But very soon we will have the opportunity of also trying this in humans. This is where a project is. We're about halfway our project, where we have this complex interaction between the different work packages. So we do hope that in the next couple of years we will be able to have a proposal for specific phages that will be able to change the fate of children with asthma. And this is our group. Thank you very much.
We are 5 minutes ahead of schedule, so if there are any questions for the three speakers, please. Do we have the cube? You have to speak very, very close to the microphone.
Hello, for the first speaker. Oh, yeah, okay. Hi, I'm a fifth-year medical student at KU Leuven. And I also did a research project about brain-computer interfaces, which is very related to your research. But I would like, the problem I saw was that a lot of data in brain-computing interfaces articles, which are about similar projects, are in different experimental setups and are too heterogenic to really compare with each other. And I would like to know, your opinion about that, and what you think about maybe forming a consensus.
I see the light. Yes. I mean, I answer. I'm the old guy, so I can answer more. I think this is an extremely important issue. Standardization in data means that you can first test different hypotheses, different algorithms altogether, which is extremely important, and comparison. You know, because a prosthetic user can use BCI, can use muscular activity, can use neural activity, and the same for quadriplegics, people with spinal cord injury. So the main question is, what kind of interface should we use for a specific patient eventually over the evolution of disease, for example, neurodegenerative disorders? The only way to do that is to create open data access where people can test and compare different approaches. Otherwise, there will be always, the scientists like me publishing a small piece of paper and with no real advance and real comparison, which should be the case. So that's something, in some cases, we are doing ourselves, for example, but clearly there is an issue in sharing data, and we should move in that direction.
Yeah, I would just want to add to that. Yeah, I think it's especially important for these like neurotech projects because you have small clinical trials. You were talking about 3 patients, 4 patients, but for a doctor, I'm also becoming a doctor, for a doctor to implement this in clinical care, you need larger clinical trials, I think. And so especially for that, I think you need to exchange data more.
Yeah, I totally agree. It's absolutely important, especially for medtech.
Okay, we have time for one extra question, please.
I have a question about the funding. Could one of you explain the difference between the type of funding you received compared to a classical EU project funding? Because we're here for EIC, to understand what the EIC can do.
Yeah, maybe I can briefly, since I'm here, tell you my experience with FET, and hopefully will be the same for the EIC. The main difference there are 2 differences in general. The submission is a shorter number of pages. So it's easier to submit and to say more focus on science. But to me, the main advantage is the flexibility that this kind of FET approach can provide. So trying, I mean, it happened to me a couple of times also in other projects to have to change drastically the kind of interface, the kind of approach. So the flexibility in following a path which is clearly high risk, it could be high paying, as in some cases you just saw, but it's really high risk. So at the very beginning, the flexibility in moving around science, changing the kind of plans you had at the beginning, it's more the spirit of the way these kind of projects are led also by the officer in the Commission, which makes a difference, in my opinion.
Thank you very much. We move now with Timo Halanti for the second part of today's session.
Thank you, Pepe. And indeed, the breathtaking pace of excellent examples of FET and Pathfinder research will continue, and we'll move now to the area of energy and environment. And the first speaker will be Antonio Marti, who is a professor at Polytechnic University of Madrid, and he's there in the Solar Energy Institute.
Okay, thank you. Okay, renewable energies are already here, and photovoltaics is actually already the cheapest way in which you can produce electric energy. However, for a massive deployment in the future of energy renewables, we need also efficient and reliable ways of storing energy, because we won't also use renewable energy when the sun is not shining or the wind is not blowing. Today, energy is more or less stored in batteries at a more or less large scale, water dams, and perhaps in the near future, it will be also stored in hydrogen. Each approach has its advantages and its disadvantages. Seems the slides are not correct. I don't know. It's okay. So in the Amadeus project, we want to add to the portfolio of ways of storing energy a new way, consisting in storing the energy in molten silicon. Why molten silicon? Because molten silicon has a high energy density. You can store in 1 liter of molten silicon about 1.2 kilowatt hours of energy, which is typically the energy you use in your washing machine in one run. This energy density is about 2 and 3 times the energy density you can store in a modern lithium-ion battery, and this is slightly below the energy density of you can put in hydrogen. Silicon is abundant. Silicon is the second element in abundance after oxygen in the Earth's crust, so you won't face strategic problems regarding its availability. There is also industrial experience with molten silicon. Actually, the picture I showed before dates from 1954, and today we melt a lot of silicon in the microelectronics industry for producing wafers, and also in photovoltaics for producing the silicon solar cells. And also in Amadeus, we are proposing a new device for converting back the energy stored in silicon into electricity. The device, we call it thermionic hybrid converter, and the idea is that it will use the energy of the photons emitted from the silicon and also the energy emitted from the electrons to produce back electricity. It's also scalable. Typically, I mean, you could store in 60 liters of molten silicon the energy you would need for between 1 and 2 houses. And in 1,000 liters of molten silicon, the energy you would need for about 60 houses. But we also have in mind a bigger picture. This is a methanol plant that we imagine using CO2, ideally atmospheric CO2, and combining it with hydrogen and using the heat stored in an Amadeus system to produce methanol. The energy will come from renewables, photovoltaics, wind, and the energy stored in hydrogen and in heat will allow the factory to run continuously, also during night, and also because of the storing capacity of hydrogen and Amadeus, it could take advantage of the electricity prices when these are the lowest. And more important, the technology is here. The project started in 2017 with designing the prototype, and by 2019, which is almost the end of the project, we are testing the first prototypes. In the picture, you can see, for example, the crucible containing the silicon, and only 4 centimeters away from this container at 1,400 degrees, a thermophotovoltaic cell operating at 60 Celsius degrees only. And I want to finish acknowledging our partners in the project, the Foundry Research Institute in Poland, the Norwegian University for Science and Technology, the CERT Center in Greece, IONBAC and CNR in Italy, the University of Stuttgart, and the Technical University of Madrid. Thank you.
Thank you, Professor Marti. And we will then move on to the next project. And it will be presented to us by Rachel Armstrong. She is a professor of experimental architecture at the University in Newcastle. This is just to really mark those who have followed the journey of PhET. You know that the interdisciplinarity and bringing different sciences together. So here we have an example coming from the area of architecture and waste management.
Thank you. Well, I'm just going to... I am the coordinator for a group of 6 collaborators on this project. Newcastle University, University of the West of England, University of Trento, Spanish National Research Council, Explorer Biotech, and Liquid for Systems Group. And our work looks at how we might make better use of household resources by using the incredible processing power or metabolism of microbes. Now, by strategically sequencing these reactions at the human scale, we can perform housework, which generates a range of products that can be fed back into the home systems, as well as being good for the environment. Our starting point is liquid domestic waste, namely urine and grey water, which acts as a feedstock. This is then passed into a series of chambers, rather like a cow's stomach. Each chamber is an ideal home for microbes. So when substances enter, they get converted into a range of metabolites, passed on to the next chamber, and so on. And these chambers are programmable and perform a specific role or function. We do that by selecting the bacterial species and also spatially sequencing these modules. Together, this is called a metabolic app. Our wild type module is based on the microbial fuel cell, which consists of an anode, a semi-permeable membrane, and a cathode. The hardware captures electrons from a bioelectrically active biofilm to make usable amounts of electricity. The cell itself processes water, dirty water, and turns it into a range of metabolites and polished water. The system's self-powered and governed by an AI that can detect the amount of electricity in each of the bricks and modulate the inputs accordingly to optimize the outputs of the system. Power is boosted by the presence of oxygen in the cathode that's provided by a gravity-fed photobioreactor with a unique geometry and triple baffle system that promotes mixing of liquid and gases. Our proof of principle system is based on a synthetic bioprocessor that allows us to ask just how far we can design and engineer metabolisms using synthetic biology techniques. The system that we've produced can metabolize nitrogenous gases, but our indicator system is inorganic phosphate, which is reclaimed from household detergents and is shown by the biosensor here on the right. To get to this point, we needed to make a number of breakthroughs. Firstly, we needed to design a synthetic bioprocessor because the hardware didn't exist before. But then we needed to figure out how we could work with microbes at scale. So we've devised the farm and labor modules within a living brick, let's call them. The farm module consists of organisms. It houses these organisms that overproduce an energy-rich substance, so they overproduce sugar. This then diffuses through the semipermeable membrane into the labor module. Now here we have a range of workhorse organisms whose genes we know very well, but don't normally live together in the natural environment. But under energy-enriched environments, we can actually get them to collaborate. So this greatly increases the range of metabolic portfolio that we can work with because we can start to design metabolisms across species. And during this process, we have identified 2 new genes which have been sent through the patent application system. We've also developed a new software called Dulix, which is a web-based DNA library design tool that makes gene sequencing more intuitive. Throughout the entire project, we've been really conscious about the social acceptability of this system because we're working with microbes. So we developed a range of modules as the project progressed that we exhibited at biennales and in international exhibitions to start a conversation with the audience about the kinds of things we were thinking and doing. This is a first prototype of the system, a living brick, which combines a vernacular brick with a microbial fuel cell to produce usable amounts of electricity. And it was shown during the Venice Architecture Biennale in 2016 and also at the London Building Centre in 2017. Our next set of modules started to combine metabolic worlds, as it were. And this is a prototype that was shown at the 4th Tallinn Architecture Biennale. And it approximates photosynthetic and anaerobic environments so they can actually exchange metabolites. These principles then informed a final design of the brick system, which is a 4-chamber system that is separated by ceramic plates. And when this is inserted into the overall apparatus, it can reclaim 100% of the inorganic phosphate entering into the system. The metabolic app is not complete without spatial sequencing. So this is the way that the system was constructed. The feedstock enters into the top of the apparatus and assisted by gravity, passes through the different brick types in the system to be collected in a sedimentation chamber at the bottom, and some of the fluids are recirculated into the system. And this is what it looks like when it's compiled. You can get a sense of the scale here, but this was not explored in a social context because of the presence of synthetic microbes. However, we did design a wild-type system for the Is This Tomorrow exhibition at the Whitechapel Gallery in London as a collaboration with artist Cecile B. Evans. And actually, it was an important exhibition because it was a follow-on from the 1956 This Is Tomorrow exhibition, which was talking about the future of art and architecture. And the work that we produced was a clear-walled apartment space so that visitors could see from the outside in. And it was occupied by phantom images, but all were supplied by a microbial fuel cell wall, which was connected to a screen-based system. So a microbial fuel cell array generated the power for some of the screens in the installation. Essentially, the work we've produced generates questions for the way that we live. And whilst I discuss some of these, I'm just going to show you a video that we produced that suggests how the living architecture system, or systems like it, microbial systems, could be implemented in practice in our domestic living spaces. So the question I think that we really need to ask ourselves and challenge is the need for 230 volts in every domestic space. And consider replacing these with 12-volt systems, which could realistically be supplied by systems like the Living Architecture Project. Now, to do this, we need innovation in low-power electronics, and we need a lot of it. We've started some of this in the Living Architecture project with the controller for the wall, which was powered by bioelectricity, and also the project developed a robot that could open and shut a window. But we need much more diversity in low-power robotics so that the living architecture experience as a domestic environment is actually a rich and interesting one. We also need innovation in biodigital interfaces so that at a glance, an inhabitant of a space can see just how their microbes are getting on, maybe like looking at a garden or finding out how their pet's doing. And we're hoping that by, let's say, creating an anthropocentric interest to the living architecture systems, that we change the relationship between human and technology from one of exploitation to one of care, so that when you buy your metabolic app, you are looking after it by feeding it with your domestic waste. And the microbes look after you by providing the metabolites that you need. And a condition of mutual care will have knock-on benefits for the environment. So in summary, the Living Architecture project is a successful proof of principle system that demonstrates our homes do not have to be sumps of obligate resource consumption, but could be sites of resource renewal and regeneration that could underpin a 21st century domestic economy, an economy of the oikos, fit for a global sustainable society. Thank you.
Thank you, Rachel. And then we will move to the third and the last presentation of this session. So I will introduce to you to Jose Ramon Galán Mascaros. He is coming from the Institute of Chemical Research of Catalonia, and he will present us the project ALEAF.
Thank you very much. I want to thank the organizers for inviting me to this session on new energy for the future, and I need to thank the previous speakers because they already set up the stage for my presentation. So I'll try to offer you another strategy or approach towards new renewable energy, and that's artificial leaves. I don't need to talk much about motivation. This is the easiest in this kind of talks. And we have a problem with CO2. You call it climate change, you call it contamination, pollution. I mean, there are many issues, and we need some renewable energies out there. But let me point to the numbers, and I think these are important. The world consumption right now is 13 terawatts. Don't ask me what a terawatt is. I don't know. But this is the world consumption, and the projections are 30 terawatts. But we have very good news with these numbers, because the solar energy reaching Earth is about 120,000 terawatts. So just looking at the zeros out there, you know that only transforming a very small quantity of the solar energy reaching the Earth, we would solve the energy problem for sure. So this is a way to go. The good news, and we have other renewable alternatives, wind is very advanced nowadays, but hey, solar, it's enough for all we need for forever, probably, looking at the numbers. But as it was mentioned before, we have a problem with renewable energies, and particularly with solar energy. We need to store it. And you can use batteries or some other stuff, and this was already discussed by Antonio. You can use molten silicon. So there are, but I think in terms of fuels, if we can do solar fuels, this has huge advantages. Sorry about that. And of course, you can go to nitrogen, but right now, the fuels that you have out there, that's gasoline, even wood, is much better than batteries, 10 times better, not only in volume, also in mass, and there's an advantage that I think is crucial. Most industries, especially transportation, are already using fuels. If we can just offer them renewable fuels, the societal transformation would be very easy. We don't need to create new items or new low energy. We can keep doing and using the same technology that we are using, but with fuels that would be renewable. Why? Because this would be following photosynthesis, but doing artificial. So we need to absorb light. We need to use this light transform it to electrical energy, and then we can oxidize water, producing oxygen, and then we can use the protons and electrons from water that is wild, available in the world to reduce protons to hydrogen, but to reduce CO2 to fuels. And we can make carbon-based fuels competitive with gasoline or oil products, but from original artificial photosynthesis perspective. And this would be a circular economy, circular energy, because then we are fixing CO2, that is a pollutant, to make fuels with the energy of sun, and then generating oxygen, and then these fuels can be immediately used, or maybe they can be transported and then generate electricity when and where it is needed. And of course, we are talking here about a CO2 generation again, but this is a circular energy system, because the same CO2 enters the system and exits the system. This is carbon neutral. We will not reduce the carbon at the atmosphere level, but we will not increase it. This is completely environmentally neutral and no effect in the atmosphere content of carbon. Okay. Well, we are talking artificial photosynthesis, and you may think, okay, can we learn from the plants? They are really good doing that. The bad news here, they are not so good. To start with, they are green. They are using only about 45% of the sun they received, and the best plants out there barely go over 2% of efficiency fixing the solar energy. They have something that it's really unique and we cannot do in technology, that they can self-repair the system. The plant's photosynthesis system is not even reliable. Every 30 minutes, the photosystem too is destroyed in the plants, but the plants can repair it. Can we repair or substitute a battery every 30 minutes? Don't think so. So we need to do better than plants. But the very good news is that we already are doing it. This is a latest example, not from Europe, but we can also do it here. You know, artificial photosynthesis at 30% efficiency has been already done in a laboratory. What's the problem? You may ask, if it's already done, what can we go? The problem is, look at the chemical symbols there. You have platinum, you have iridium, you have gallium, you have indium. You cannot do an upscaling of these kind of systems. So the problem is not only technological, the problem is also about the costs. Because I am dreaming high, okay, I think we can do it. I think we can reduce the cost and be competitive with fossil fuels. It's out there, you know, fossil fuels are very cheap, but if we do it right, we are only needing sunlight, CO2, and water. We should be able to make it simple at low cost. And this is really the challenge that Alif started 2 years ago. We are in the middle of the project. So we have 8 countries, 13 partners, 9 universities, but also 1 industry. The industry is special to give us the knowledge of what is available, what is scalable, because we want to build a photoelectrocatalytic device that will take CO2, water, and sunlight, nothing else. But we want to make fuels with this device without any external power. And the thing is that we want to do it economically profitable. Is it possible? Is it doable? This is what we want to do. And that's why we are only using earth-abundant materials, low-cost materials. We are doing scalable processes because we want to compete with fossil fuels. We just not want to do a project that is just scientifically relevant. We want to move forward and to do a project that is viable, affordable, and scalable. Of course, we may fail. Maybe our device is not economically relevant enough, but still I think it's important to reach this goal, that our success goes to advance the field, to increase the critical mass, to bring society the news that this is doable, let's make it cost effective, and also to consolidate the renewable energy vectors. And of course, such a challenge needs a really a large effort, and these are the team of all our institutes and universities and Covestro, and of course the FED support to bring this project to fruition. So if you want more information about our project and our progress, yes, you can check out on internet, Twitter, LinkedIn, and Facebook. Thank you very much.
Thank you. Thank you very much. I think we have some time for questions from the audience if you want to clarify or comment something that you just saw in these 3 projects. I think there is the microphone there. Everything is clear. Anyway, I would like to make one comment myself. I think if you look at these, not only these 3 projects, but also the 3 ones that we showed earlier, okay, it's interesting and we'll get a glimpse of the tomorrow's society, perhaps through these future technologies which are coming out. What is my point is that I hope you can see the red line, which is common to all these 6 projects, actually. And I think some of the speakers referred to this as well. Yes, we have interesting proof of principle of a technology, but the underlying vision is that in all these projects, there is a kind of an idea of a tomorrow's technology, which is as everyday as this microphone or a telephone or whatever, which is very relevant for the society. I mean, in each project, you have something which is a very big problem or challenge of everyday life. And they are trying to solve that in their own corner with a view of tomorrow's technology. But what is interesting to note that by no means that technology will be a ready-made product at the end of these FET open projects or FET proactive projects. What we need is a follow-up. And that's why we are bringing now the FET together with the EIC, because we have seen that if we don't have a follow-up, there is a huge delay. People will need to go back again and, let's say, try to find the funding for the next phases for something which we already know that it's excellent. We have followed these projects over the years. We have followed their publications, so we know that there is an interesting technology coming about. So the point is, why do we have to go and leave them on their own and hope that somebody picks up the ball from the floor? And so that's the whole point of the EIC, which we are talking about here, that we are linking FET, which is becoming now a Pathfinder, together with the accelerator phase, where we don't lose time in putting these technologies to the market and the society. Now, if you are interested in continuing and having a look, and there was a parallel session next door as well, maybe there is a recording for that, but there are several hundreds of this type of projects ongoing at the moment, and we have encouraged them to do this week. I think there is a slide about an event which is ongoing virtually on Future Tech Week, and we have encouraged all our projects to do publicity exercises this week. Many of them organize events or they are active online. Please watch the space and be active in following up on these projects. With this, unless if you have come up with a question while I was speaking, I would like to close the session and thank you for the speakers and the participants for this one. And let's see you around during these RNI days. Thank you.